All right, what we want to do here is we want to go over the labs. Excuse me, I'm going to give you a lab test on Monday over the labs. And
we have a psych exam. Maybe psych next Monday.
You do? We'll have to figure it out for you then. We'll make a special schedule for you. The best way to study for this lab exam is to go back over all your labs. You should have your lab book and your log book. And you can use your lab book to look over these things. Now, lab 1 we didn't do in here. You got your lab book out? Let's look at lab 1. It's on page S17.
Can I get mine? That's not one.
Yeah? Let me just tell you what happens in lab 1, although we didn't do it, just in case you see a variation of it or something. It's an evolution lab. And what you do in lab 1 is you grow up a bunch of plants. And then when the plants get big enough, they've got flowers on them. And then you take a little paintbrush and you mate one type of flower with another type of flower. And what you do is you choose the flowers you want to mate. So what you could do is you could grow all your plants up and take only the tallest ones and mate them with one another. And kill all the short ones. And then after you made them, the flowers produce seeds and you plant the new seeds. Are y'all following me here? And the new plants will grow up. And what do you think will be about the new plants that's different from the old set of plants?
They'll be more tall.
They'll be taller, don't you think? And don't you think if you took in the second generation the tallest of those and made it with the other tallest of those, the next generation will be even taller? And that's what you're doing in that lab. You're taking the average height of each generation and seeing if you can move it in one direction. What do we call that type of selection? There's a name for it.
Yeah, it's artificial selection.
Artificial selection is where you pick the organisms that you want to make so you can breed a certain trait.
What's the other kind of selection?
We didn't do that lab. It takes about 3 months. And I decided not to do it. We had so much other stuff to do. But basically, that's what it is. It's showing that artificial selection can happen.
Are you gonna test us over the labs that you didn't do?
Probably not. No, probably not. I will give some general lab questions though that they've had on the AP exam that are kind of not exactly like any lab that we did. But you have to use your thinking of what we did and apply it to a new situation. Do you see what I'm saying, Emery?
Yeah.
We have to know these really well.
For the lab test?
Yeah, it'd be good to know these really, really well.
Is it just like a written test, or is it like we're actually in the lab?
It's a written test. There's multiple choice in there.
So over all the labs we've
just over lab stuff. Labs we've done. And then there'll be some on another lab that nobody's done.
Is there an essay?
That you have to figure out. Yeah, there'll be essays and multiple choice.
On the lab thing?
On the lab test.
But it can only last one period, right?
It'll only last one period. Okay, the second lab was Hardy-Weinberg stuff. Do y'all remember the Hardy-Weinberg equations? And how to do those?
Plus 2p2 plus q squared equals 1.
That's right. It's on your sheet here. And that you'll have. You don't even have to memorize it, but it's not that hard to memorize.
Wait, so we'll have the sheet?
You have this sheet on the AP exam. We went over this on some of the days you weren't here. Right there, this Hardy-Weinberg equations, that will be on, you'll have this sheet, and there are the equations. So, I have the Hardy-Weinberg problem sheet. I've already given you this, but if you want to try it again, the answers are on the back. So if you're not too sure about Hardy-Weinberg, does anyone want one of these? A Hardy-Weinberg practice problem sheet? Sure. I'll just give it to you, and you decide later if you don't want it. And that's really what Lab 2 is about. That big computer program we made, the Excel program is really a program that breeds populations and gives you new P's and new Q values and such. And it kind of figures it for you. So it was to kind of get you comfortable with using these numbers. Let's try the first question right now. Okay? In Drosophila, what are Drosophila? Do you know?
Fruit flies.
And fruit flies.
Yep.
The allele for normal length wings is dominant over the allele for vestigial wings. In a population of 2,000 individuals, that's our population, 315 are recessive. How many individuals would you expect to be homozygous dominant and heterozygous for this trait? Always in questions like this, you want to figure out the percentage that is homozygous recessive. If you ever figure out the percentage that's homozygous recessive, you can work from there. And use the equation to figure everything else out. Again, the equations are P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. Okay? So, they say 315 show the recessive phenotype. So what percentage is the recessive phenotype? That's what we want to get to first.
15%.
15.75%? Let's call it 15.8%. How they got that was 315 out of 2,000. 315 divided by 2,000 will give you...
Times 100.
Yeah, times 100. Y'all see? So 15.8% or 0.158. 0.158 is the same as 15.8%. So that's our Q squared. Q squared equals 0.158. So once you figure the recessive percentage, you have Q squared, and you can figure everything else out from there. So always go, if they ever give you any problems, always try to figure out the recessive phenotype percentage. And that's usually what they should go with. So what's Q? If Q squared equals 0.158, how do you figure Q?
Square root it.
You square root it, which is?
0.397492.
So Q equals 0.397. That means P equals 0.603.
Yep.
State championship math team. Gosh, I'm so great.
Well, you told me to do multiply it times 100, so why did you do point?
Well, if you 0.158 is the same as 15.8%.
I know.
So if it's asking you the percentage, I mean, you could. It depends how the question's asking it. If it says what's the percentage of recessive, it's 15.8%. But to do the math, you have to use the decimal point. You see how we got P here? You see how we got Q? Yes? So now, P squared is my homozygous dominant, and 2PQ is my heterozygous. So if I want to figure P squared, I just take P and square it. I bet it's 0.36 something.
0.364.
364. And Q squared, we already know, is 0.158. We figured that out earlier. So this one is 1 minus that minus that. Or 2 times that times that. What's our percentage there? 36, 46, 52, 48?
40.479.
That's 47.9.
So 36% about are big A, about 48% are big A, little a, and about 16% are little a, little a. But remember, the population is 2,000. So 36% of 2,000 is 721, right?
720.
But 36.4.
Oh. Or 720.
You didn't see how we got this?
No, 17.
You don't see how we got this? Because there's 2,000 in the population. So if 36% of the population is big A, 36% of 2,000 is what? You do 0.364 times 2,000 and you get the number.
That's 728. Alright. 2,000 times 0.364.
Oh, is it 728? I'm
Yeah.
sorry. Good job. State Championship math team let me down there. So what percent? So what's the heterozygotes? How many?
956.
What is it?
956.
956.
I got
958.
958.
So a good way to think about this, if you have problems with numbers, Stephanie, if I asked you what 50% of 2,000 is, could you get that pretty quickly? 50% is half, right? What's half of 2,000? 1,000. So if you have 47% of the population, you know that's got to be pretty near 1,000, right? Because 50% is exactly 1,000. So there we are. We're pretty near 1,000. You can kind of check to see if your answers are right. Go.
Real quick. 0.158 times 2,000. Can you give us the number? That's gonna add up.
0.158 times 2,000 is.
Well, we're gonna add up to 2002. That's gonna be our number.
Well, because we've rounded everything up when we were doing this.
360.
360. Yeah, we've rounded off. So every time you round, you get away from the exact number a little bit. If you weren't doing any rounding, you'd be right on it. But you would have half individuals, which is really no such thing as half an individual. Make sense? Try the rest of them. If you're not good at Hardy-Weinberg, try the rest of these. Again, I put the answers on the back there for you. There will be Hardy-Weinberg questions on this lab test. Your questions, making sure that you understand the equations. Investigation 3. Lab 3 is where we went on the computer. This was kind of fun. And remember these cladograms, and you took the DNA from this, they said they found DNA, some genes in this little dinosaur-looking creature. I don't know what it was. And they had the genes, and you downloaded the genes, and then you plugged them into a website, and that compared the genes that were in the fossil to all the genes in their database. Do y'all remember doing that?
That was hard.
And it gave you matches for the genes that you found from that fossil. And after you did them all, you found out from that fossil that they had found there was a gene from, I think, a chicken. You remember that? What else were the genes from? A zebra finch, which is a bird.
Yeah, crocodile or alligator.
Alligator gene. And so,
Part bird, part reptile. So.
wasn't there an insect gene
Yeah,
in there too?
fruit fly.
A fruit fly gene. And so those genes give you kind of some evidence on where to put it in on this little family tree here, this little cladogram. And most of the genes were crocodile/bird up in here, so you probably put it up in here, although there was one insect gene. But insects came before these guys, so it could have still some insect genes in its genome. And so basically this was just working with computer database. Do y'all remember looking up your DNA alias? Did anyone do that little part of it? Where you took your name and you spelled it out in DNA code, and then you looked up. Stephanie, you're nodding. What were you? Do you remember?
I'm not sure.
I was a yeast.
I was human.
You're a human? No way, really?
I was a mosquito.
Mosquito?
Nobody would tell me how to do it. It's like,
Oh, well, I know.
So look back over it. We call that computer program a blast. And what they'd probably do, the kind of questions they'd probably give on this, is they would say, here's a gene sequence. You blast it and you get these results. You know, how do you interpret this evidence, or something like that? I really don't know how they're going to ask. I'm just trying to guess. They've never. This is all new, even for the teachers. We've never had any test over this stuff, so I'm not sure how they're going to quiz you on it. But if you understand DNA and how it functions, you're good.
I was a nitrogen fixing bacteria.
Nice. Yeah, living in a lagoon, fixing some nitrogen.
Yeah. So the genome, the Gattaca, that's something. Those are the first genes.
Those are just letters of DNA, and
yeah.
the movie's kind of about DNA.
But isn't that significant?
you know what movie is about?
Gattaca?
Yeah, the GATT.
I don't know that it's significant.
Okay.
Have you ever seen the movie Tuxedo with Jackie Chan? It's like about like the water strider and like
Tuxedo?
tuxedo. And he like it's like this tuxedo that takes over your body and like do weird things. But
With
like
Jim Carrey?
It's called The
I
Mask.
don't remember. But like it's like the water strider. He thinks he's saying Walter Strider, and then he figures it out. He's like, oh, Walter Strider, and it's like a water strider, like the spider that makes it do all the stuff.
Okay,
It's pretty cool.
so how does that relate to?
I don't know. I don't know how I thought about that.
I thought you were gonna pull out a DNA.
I have no idea.
I have seen that movie. I think it was Jim Carrey that was in that. It was funny. Okay, Lab 4. This is a lab about osmosis and movement of water. There's a few different things we did in this lab. We took those little cubes. You remember those colored cubes? And we put them in vinegar. And the vinegar starts diffusing in. And the larger cubes took a lot longer for the vinegar to make the cube completely clear. And the smaller cubes got clear really quickly. Y'all remember that stuff?
The surface area to volume.
Surface area to volume ratio.
Which
You want
never
it
makes
big.
sense to me.
Which never made sense?
Yeah.
If you are a very big cube, you got a whole lot of volume on the inside.
Okay, I got it.
Little cube, hardly any volume, mostly surface area.
The way
I
I
forgot
remember
the
it,
volume
that so the way I remember is that in area you square your number, but
Volume
volume.
cube. The volume's going to go up faster than area. That's a good mathematical way of remembering it.
Yeah, just think about that.
So that was the first thing. The second thing was taking the little baggies. Remember the little baggies? And we put sugar inside it. And dialysis tube baggies, and they have little holes that can penetrate, and the water can go through, but the sugars cannot. And so if I took a little baggie, and I'm real good at drawing little baggies, and I put it in a beaker, that's a beaker, and this is water in a beaker, sorry, I don't know what that is. It's a weird melted beaker. And inside the bag, outside here is just water, H2O, pure water. And inside the baggie is sugar water, 0.4 molar sucrose plus water. We have some words here you were supposed to know. What would we call the bag, the solution inside the bag in this situation? We'd say inside the bag is hypertonic. And I always remember hyper has more sugar in it, or more salt, or whatever the solute is. So the side that has the most solute is hypertonic. The other side is hypotonic. Hyper and hypo. If you have a hypertonic, you always have to have a hypotonic. One's going to be hypotonic. If I made this outside 0.8 molar sucrose, and inside is 0.4 molar sucrose. Now all of a sudden, outside is hypertonic. Because this has more sugar than the inside. The inside is now hypotonic.
It goes from hyper to hypo. Then?
Yes, it always goes from, water will move from where, well, I'm sorry. Sugar would want to move, let's go back to our original situation here. Whoa, excuse me. In this case, the sugar would want to go outside the baggie. But it cannot, because the holes in the baggie aren't big enough to let the sugar through. What's water going to do?
It's gonna go in.
Water will go from where it is in high concentration to where it is in low concentration. From where it is plentiful to where it is scarce. Water always goes where it is scarce. And it is more scarce in the bag than outside. So water, what's going to happen since the sucrose can't come out, the water will go in and our bag will bulge up with water. And do you remember what we did was I had 5 different colored solutions that were all different concentrations, and you had to figure out which colored solution had the most sugar. Do you remember that, Savannah? Are you cold?
A little bit.
You want me
That's good.
to adjust? Are you sure?
Yeah.
How about a jacket?
I put them out.
Maybe those little hand heaters? I got those for golf. Still have a few.
Okay.
One of those? No?
So thank you.
Those are nice. Okay, so the water will go into this bag. What will we say if inside the bag and outside the bag were exactly the same?
Isotonic.
Isotonic. And if we were going to give a water potential number, do you remember the little psi sign?
Oh, yeah.
You remember that?
Negative ICR.
That's water potential. And you can figure out the water potential by using a formula. And they give you the formula on the page. It's right there on the right side on the back of this page. Solute potential equals negative ICRT. I is a constant. C is the molar concentration, which in this case is 0.4. R is a pressure constant, which it lists there. And T is the temperature. What if I said that this was happening at 273 Celsius?
Does it have to be in Kelvin?
273K, I'm sorry. Could you figure out the water potential if I gave you it was 273 Kelvin?
Mhm.
That's about room temperature. No, wait, that's not
That's
right.
freezing.
273 is freezing. Let's not do it at freezing. That means we have ice. Let's do 293. That's about room temperature. And the water potential equals negative ICRT. Anyone with a calculator can do that for me real quick?
What's the I?
I is the ionization constant. It's 1 because sucrose does not ionize in water.
And what's C?
C is the molar concentration, which is 0.4.
What is R?
R is 0.0831.
And T is 290.
T is 293.
Got negative 9.74.
Negative 9.74. And what's the water potential of the water in the beaker?
0.
0, because it has no solute in it. And so it's going to run toward the negative. Water always goes from high water potential to low water potential. Listen, study up on tonight on labs 1 through 4, although we didn't do 1. So 2, 3, and 4, and we'll continue talking about the labs tomorrow.
Can I take a movie first?
Yeah.
Can I take one as well?
move it, and move it. Did you return your movie?
Yeah.
Oh, my
So, this one here, got two first place stickers. Unbelievable.
Do I get a sticker?
I don't think, I've changed the rules.
I got
Jeff.
third
You can only
place, I
run
got
on
stickers.
Friday.
Oh yeah, here's a sticker.
Mr. Willis, do we get stickers?
Because I want y'all studying during the week. I changed it.
Alright.